Hello everyone, thank you guys for liking my channel, thank you guys for watching my videos. I appreciate all you guys' thumbs up, all you guys' comments. I've never done this before. I'm going to actually put together every single component known as cellular respiration. This is so important for you guys who study introduction to biological science, microbiology, or who are trying to get into biology at a college or university level. Never have I seen a video like this before where you can actually contact a person through email, cell phone, and I will respond to you. I will be there every step of the way to help you guys out understand this. Let's start off as the best way we know mankind. So what I'm going to do right now is put in glucose. For those of you guys aren't familiar, glucose is going to be a 6-carbon. Now I'm going to put glut, and then I'm going to put back glucose 6P or 6C. And then from here, I'm going to explain something. It has to go through the glucose transport before I can get into the cell and the cytosol. And this is going to take place in the cytosol, so it's not going to be aerobic, it's going to be anaerobic. And then you have an enzyme here called kinase. And the specific name is hexokinase. And then it goes into what is known as glucose 6P. And the reason why it's going into glucose 6P is because it's going from ADP into ATP because of the enzyme hexokinase. Then it becomes known as fructose 6P. And there's an enzyme known as isomerase, phosphoglucose isomerase, which doesn't require energy. And then it goes into another enzyme, the ATP becomes ADP, gains an electron, phosphate, and then this enzyme is called kinase, which is going to be phosphofructose kinase. And then from here, big money and big bucks, if you guys understand this, big bonus. This is called a triangle. It's going to have phosphofructose lyase, which is aldose, which is going to be the enzyme known as lyase. And then from there, it's going to split apart. And let me try to explain this the best way I know can. 2-glyceride hide. And it also becomes 1-dihydroxysteine. And there's an enzyme in between these two called TPI, triose phosphorus isomerase. Triose, remember you guys, triangle. And then from here, it goes into a position, not this one, but the 2-glyceride hide, goes to a dehydrogenase known as a G3P dehydrogenase. This could be the enzyme, where it actually goes from NAD plus to NADH. And for those guys who aren't familiar with NADH, NAD plus grabs like a net the hydrogen, and then it becomes super NADH. This is more superior than the ATP, has more power than ATP. And then from there, this could be known as 1,3-biphosphate glucose. And this is kind of like Code to the Island, how it goes from 1,3, then it reaches the enzyme, then it goes into known as 3-biphosphate glucose. And this enzyme is going to be mutase for M. Then it goes to another enzyme. Oh, I apologize. This enzyme is going to be a K. Then it gets 3-biphosphate glucose. Then it goes to another enzyme, which is going to be a mutase. And then it becomes 2-biphosphate glucose. Then it's going to be known as another enzyme known as a lyase. Then it's going to become known as 2-phosphoglyceride. And then from there, it's going to becoming then it's going to go from 2-phosphoglyceride to 2-phosphophenol. And then that's going to be going to a pyruvate kinase enzyme, and that's kinase. Then you have here 2-pyruvate. And I'm going to spell this out, and I'm going to spell it out 2 more times because this is important. When it goes from glucose to pyruvate, this is known as glycolysis. So this would be known as glycolysis. So you got a bonus right there, glycolysis. Okay, it's in cellular respiration. Now, this pyruvate is a 3-carbon molecule, so 3 carbon, 1, 2, 3. It's going to be entering the mitochondrial membrane, and it enters the mitochondria through something called a pyruvate dehydrogenase, which a carbon comes out. So now it's just 2 carbon. So it's just 1, 2, 2C. Also, NAD plus becomes NADH. And then this 2 carbon is known as acetyl. And also, one thing I want to mention too, you're going to receive something here known as coenzyme A plus SH. And this is going to be going in between the and it's going to be known as the citrate. This enzyme is going to be known as the citrate synthase, and this part is going to be known as the oxaloacetate. This right here is a 4-carbon molecule, so 1, 2, 3, 4, 1, 2, 3, 4. For those of you guys aren't too familiar, if you guys want to add a bit more, there's something called Pyruvate Carboloxanes, and what's going to happen over here is that the ATP becomes ADP. So basically, this is going to be called pyruvate carboloxanes. This goes to oxaloacetate. This is 2-acetyl coenzyme SH, and then it becomes coenzyme SH over here. And then this right here, the 2 acetyls going in between the oxaloacetate and the citrate. So citrate right here is a 6-carbon molecule, 6C. It gets 2 from the acetyl and 4 from the oxaloacetate, and then it becomes 6 carbon. From there, it becomes cis-aconitine, and this enzyme is going to be known as aconitase, where an H2O comes out, and then it goes through aconitase 2, where H2O comes in this time. Then you get to something known as the isocitrate. And then from there, it's going to actually go through an enzyme known as dehydrogenase, isotope dehydrogenase, where you lose a carbon. So you're going to lose a carbon. And then you reach something known as ketoglutarate. If you guys try to remember ketoglutarate. Ketoglutarate sounds like karate. So expect to get beat up. Expect to lose a carbon before, and expect to lose a carbon after. So try to remember ketoglutarate. There's something called sunico. And then from there, big bonus, you guys. You're going to earn some money if you can recognize this one. This is going to be known as complex enzyme 2, the only enzyme known to mankind to be in the Krebs cycle. So then from here, you're going to reach formaldehyde, and then from here, you're going to reach malate dehydrogenase, and then you're going to get into the oxaloacetate, which right there, you got the sunic, you got the the fumarate enzyme, and then you have the malate. This could be known as the malate, and then after the malate goes to the malate dehydrogenase, which becomes 4 carbon. So you lose a carbon here, and you lose a carbon there, you lose a carbon before. Ketoglutarate. Remember, ketoglutarate. No sound effects. Oh, maybe sound effects. Bah. Ketoglutarate. Here you got the electron transport chain. Enzyme 1. NADH plus. H plus becomes, and let me see if I can do this correctly, NAD. Complex enzyme 2. This is going to have the C. This is going to be the letter Q. Coenzyme C, coenzyme Q. Then you're going to have complex enzyme 3. 4 hydrogens. I want to see those 4 hydrogens, 4 H, that are going to assist in the transportation. And big money for this one, if you guys can understand this one. Complex 4 is the enzyme, the only enzyme that takes the oxygen from cellular respiration and turns it into H2O. So that's going to help you guys out. Remember the cellular respiration. And here you're going to have the ATP synthesis. ATP, remember, this is with ATP synthesis, the energy. So we can call this one right here Roman numeral 5. I'm going to try to help you guys out real quickly. Big stuff to remember as of right now. Try to understand that glucose gets turned into pyruvate because of the enzymes. 3 carbon. This right here is going to be the mitochondria. And then inside the once it gets to the mitochondria, it goes to the pyruvate dehydrogenase where it becomes 2-acetal. This Krebs cycle, TCA cycle, citric acid cycle, it's inside the mitochondria, it's inside the matrix. The electron transport chain is inside the inner membrane. So let me see if I give you guys a good review. Glycolysis starts from glucose, enters the cell. From glucose, it's going to be dissolving in the cytosol, the chemical dissolvement. Then it turns into pyruvate. Pyruvate enters the mitochondria. From a 3 carbon turns into 2 carbon. That goes to the Krebs cycle. It loses a carbon before the ketoglutarate, after the ketoglutarate. Then you're going to have the electron transport chain. That's going to go from complex enzyme 1, complex enzyme 2, complex enzyme 3, complex enzyme 4. And if I need to make this more sense, I'm just going to write it in right now. The cellular equation right now for us, and I'm going to try to do this as neatly as possible right here. We are animals, so we are known as 6O2 plus 12H, 6C, 6O, which is glucose. If you give an animal cell 6 molecules of oxygen and glucose, which is right here, in return, you will get 6H2O, which is located right there, plus 6CO2. And let me see if I can help out with this. So oxygen's right here, going to complex enzyme 4, coming out in the H2O. And then you have the glucose, which enters the cell and is in the cytosol. Then it gets modified to pyruvate. It goes into the Krebs cycle. And from there, you can see through the Krebs cycle, you are receiving your carbon throughout this process. So this cellular respiration, unlike some of the other videos, I want you guys to understand this. I got a profile up there. I'm keeping it very public and open to the community all over. I want you guys to ask me any questions. Unlike everybody else on YouTube, I'm going to put my information out there. I'm going to put my email, my phone number. I'm here for you guys. I'm here to support you guys. I hope you guys enjoyed this video. Let me know if you guys have any questions or comments. And please pay attention and watch my other videos. I'm here to support you guys. Thank you very much for listening.